Hello everybody, it is embryology time. It is week 10. This is the last embryology lecture of the quarter, and it's winter of 2021. Here we go. So where we left off, we just started to differentiate the mesoderm layer into three layers. We created a paraxial mesoderm, an intermediate mesoderm, and a lateral plate mesoderm from the run-of-the-mill mesoderm cells. How do we do that? Basically, the paraxial mesoderm was exposed to low levels of BMP. Intermediate mesoderm was exposed from above. These cells above are secreting BMP, and it's soaking down into these cells and turning on genes so they can create their own BMP, but they don't. They create as much as the cells above, so they kind of fall in line. Like these cells secrete low levels of BMP, so do these. These cells medium levels of BMP, so do these. Paraxial mesoderm secretes very high levels of BMP. Okay, that's where we are. We also, at the same time this story was going on, we've already talked about how making a neural tube, but we have to back up a little to talk about this story because the neural plate was just forming and hasn't started folding yet. Nodal cord is still pretty much rules the area. Right, so let's get into this lateral plate mesoderm because we're going to split this into two layers basically. In a nutshell, here's actually where we're going. So we're basically going to split this and create a cavity right here. And we're going to kind of extend these layers. So that's where we're headed right now. All right, so lateral plate mesoderm, some AKAs, lateral mesoderm, lateral intraembryonic mesoderm. Usually just when you say lateral plate, that means that you don't have to say the word intraembryonic anymore. Because we have to be careful, right? Because there's an extraembryonic mesoderm. There's a splanchnic and somatic layer of that. Well, we're going to have a somatic and splanchnic layer of the lateral plate mesoderm, but we don't have to call it intraembryonic because there is no lateral plate extraembryonic mesoderm, in case you were wondering how that worked. So paraxial mesoderm starts forming on day 17. Again, lateral to that, the intermediate mesoderm, we just discussed this already, will form. And yeah, it doesn't segment like the paraxial mesoderm, forms into blocks, somites, which we're going to talk about here in a little while. There's the lateral plate, there's the intermediate mesoderm. It's kind of review still. So how does it form? We already said this as well. We remember that BMP4 soaked into it and turned on BMP4 genes, a lot of BMP, high levels of BMP soaked into it. And because the cells don't secrete the BMP4 inhibitors, noggin, chordin, and Cerberus1, which is called CER1, there's nothing to inhibit BMP4, so very high levels of exposure occur. We kind of already said that, didn't we? I could probably take those slides out of there. Can be classified into cranial and caudal regions. So the cranial and caudal lateral plate mesoderm is a little different. I'm not going to go crazy into that. The dividing line is about the 5th somite, which will make more sense here in a little while. And it's based on more of the gene expression than anything else. But the cranial region of the lateral plate mesoderm goes on to form a lot of the heart stuff, as we'll see, as well as the dorsolateral neck muscles, whereas the caudal lateral plate mesoderm ends up forming the limb bud. So that's incredibly important. It's not the only thing, but it's the major contributor, the former. Its mesenchymal cells swim in and create basically your arms and legs. So a lot of stars here. So what creates the limb buds? It could say lateral plate mesoderm. If the question is really easy, it'll just say mesoderm. Right? It all depends how deep these questions go. But that's very, very important. Of course, the heart, not to say the heart's not important, right? That's formed by this lateral plate mesoderm, so it's quite important. In fact, we're not going to get this far. Make sure you watch my video online, though I'm, of course, not going to test you. I have like way more lectures than I have time for. But we didn't talk about folding, but I have that online. You can go see folding. But as folding occurs, you start popping up the arms and legs, and they pop up as these little buds right here. And yep, primarily derived from lateral plate mesoderm, so important stuff. So we have a solid lateral plate mesoderm, and about day 20, it's going to form little cysts in it. And those cysts, kind of like the same story which happened to the extra embryonic mesoderm. Remember, we got cysts in that? That was one tissue. And those cysts coalesced into a cavity called the chorionic cavity. Well, it's the same kind of deal here. We have some little cysts. They're called coelomic spaces, or coelomic vesicles, or intercellular clefts, which Larsen calls it, depending on which board book you use. So I don't think that's super important, because they really don't do anything other than coalesce into one big cavity. And yeah, beetles come together. Those little coelomic spaces come together and form a intraembryonic coelom, or Langman calls it the embryonic body cavity, which I don't like because it's not a body cavity yet. But that's Larsen, or that's Langman doing his thing, kind of being an outlier, and makes it frustrating for students when there's so many different a.k.a.s they have to learn. But nevertheless, Langman calls it the embryonic body cavity. Lateral folding is also starting to occur about this side time, but we're not going to talk about that. So here's where we are. So those little cavities coalesced into a big cavity called the intraembryonic coelom. Okay, great. What happens? Who cares about that? Why did I even bring that up? Very important stuff here. So these cavities, this intraembryonic coelom, the tissue in that during the second month of development, it forms the big 3 P's, the big 3 P cavities: pericardial cavity, pleural cavity, peritoneal cavity, or peritoneal, tomatoes. I like peritoneal, actually. I think it's more officially pronounced in the United States, peritoneal or peritoneal cavity. I like peritoneal. More England is pronounced more like that. But anyway, yeah, so that surrounds the heart. The pleural cavity surrounds the lungs. Peritoneal cavity, that's your gut cavity, surrounds all your intestines. And you'll learn all about that next quarter in Gross 2 Anatomy. And we'll talk a lot about these in 5th quarter when we talk about pathology of this stuff. All right. Here, I threw this in here. This is so important. So this is the sac around the heart. That's one of the 3 P cavities. And it's actually made up of 2 layers. There's an outer layer called the parietal serous pericardium. There's an inner layer called the visceral serous pericardium. It's often called the epicardium, which is great. These are epithelial cells that have the ability to secrete serous fluid. Makes up a serous membrane. And you normally have some slippery stuff in here, so when your heart beats, it doesn't rub friction against this really thick outer fibrous pericardium. Also note that the great vessels here, especially the ascending aorta, it's sealed up here, so this part of the aorta is inside this pericardial cavity. And so if you get a DeBakey 1 or 2 aneurysm, or a Stanford A dissecting aneurysm, it can leak into this cavity and fill this up with blood, and it can strangle the heart. That's called cardiac tamponade. I'm digressing though. Let's get back to embryology. So what happens next? So our little cavity in the lateral plate mesoderm, it keeps on developing, and actually rips right out and communicates with the extraembryonic coelom, or chorionic cavity, whichever you like there. And because it rips, now we have 2 new layers that actually match the extraembryonic mesoderm. Remember the extraembryonic coelom, or the chorionic cavity, way back before the I don't know what week, maybe week 3 or 4, we talked about this, how this is the extraembryonic mesoderm was split by this same deal. But now we have mesoderm right here. This cavity is going to sink right in here and meet the lateral plate mesoderm. I'll show you what I mean. Here's a better picture. So now this looks better. Back to that same kind of head-on view. So now we have ripped our intraembryonic coelom and it's communicating with the chorionic cavity for a while, but until folding occurs. And then these layers take on similar names. This was the splanchnic extraembryonic mesoderm. Some people call it the visceral mesoderm, like chiropractic board books. This was the somatic extraembryonic mesoderm, remember? Some call it parietal mesoderm, and different combinations of those. But these take on the same names. So this is going to be a somatic lateral plate mesoderm, and this is going to be a splanchnic lateral plate mesoderm. So the names follow suit. So it's simple as that. Now we just created two new layers there. All right, so as I just said, the two new layers are somatic lateral plate mesoderm. Sometimes it's called parietal lateral plate mesoderm. So you have to watch out, be very careful when you're answering questions. All right, everything we already said. Splanchnic lateral plate mesoderm. Everything already said. Now the combination of the endoderm and the splanchnic mesoderm, I'll just call it splanchnic mesoderm for short. Or I should call it splanchnic lateral plate mesoderm, really, that should be called. Because splanchnic mesoderm might be extraembryonic mesoderm. But anyway, those two make up something called the splanchnopluric mesoderm, the two layers combined, or splanchnoplur. So what is that? So if you combine these two layers right here, so there's ectoderm, and if you combine those two, that's called somatoplur. This one's called splanchnoplur. Got it? All right, and there's our two layers again. There's our somatic lateral plate mesoderm and splanchnic lateral plate mesoderm there. And it's meeting the splanchnic lateral plate mesoderm morphs into and meets the splanchnic extraembryonic mesoderm. See how that works? Splanchnic, splanchnic, somatic, somatic. Got it? Oh yeah, the a.k.a. nightmare. Come on, embryology authors, have a conference and get on the same page with this stuff. It just makes it so hard for students. And they wonder why everybody hates embryology, because the authors can't even agree on what to call stuff. Nevertheless, so just watch out for parietal mesoderm. That's a chiropractic book. They shouldn't call it parietal mesoderm. It should be called parietal lateral plate mesoderm more. And because it's confusing. I teach this. I've been teaching this a while now. Because I think, well, some authors call parietal mesoderm, that's the parietal extraembryonic mesoderm. So just a big mess. I'm going to stick to somatic lateral plate mesoderm. I'm always going to use lateral plate mesoderm when I'm talking about the mesoderm in the germ layer. After folding, what do these things form? Lots of stars here, right? Boards love to know what this stuff forms in the adult. And so the somatic lateral plate mesoderm, now we're getting even more specific. We already said lateral plate mesoderm forms the limb buds, but let's get even deeper. It's actually the somatic lateral plate mesoderm that forms the limb buds and also the lateral ventral body wall, not the ribs though. The splanchnic, which is in closer, that's going to form the mesentery. The big one is the gut tube. It's going to form the gut tube. The gut tube, some authors will say the splanchnic lateral plate mesoderm forms the respiratory tract. That's not, I mean, it's true in a way. That's where it comes from. But the respiratory tract buds off of the gut tube. So I should put gut tube instead of GI tract here. Gut tube it should be, which forms the gut tube. Oh, I did right here, embryonic gut tube. And that's the GI tract. But the foregut actually turns into the respiratory tract and the digestive tract walls as well. Now, I don't have time to go into it, but this is really important, this whole process of folding. So there's the link, and just you can YouTube Dr. Gillard folding embryology, and it'll pop this thing right up if you're in YouTube. But it's important to understand how the kind of the flat tri-layer disc folds into a structure like this and creates all this stuff. But I don't have time now to talk about that. So let's talk about now, let's leave the lateral plate mesoderm because we've kind of made it similar to the extraembryonic mesoderm. There's a third wave of cranial migrators. Remember, the first wave went through the nodal cord and became the precaudal plate, and then, or went through the primitive node and became the precaudal plate. A second wave came through the primitive node and became the nodal cord. Now there's a third wave that comes through of these epiblast cells, and these migrate a little bit more lateral, and they're going to form something different. Interesting about this third wave, they don't go through the primitive node of the primitive streak, which is a little weird. They stay lateral to the developing nodal cord, and they morph into something called the cardiogenic mesoderm, or cardiogenic area, or cardiac, these AKs drive you crazy, don't they, or cardiac crescent. So it's got to have one of these AKAs in here. Cardiogenic area, I mean, that's a good one. Cardiogenic mesoderm. Crescent is nice because it looks like a crescent. So it surrounds the precaudal plate. Let's look at a picture of what's happening here. So we have cells not going through the primitive node, but we have cells migrating up like this. So they're anterior migrators, or cranial migrators, but they stay a little bit more lateral. But they form this plate, and this is going to become the heart, which is another thing I don't have time to get into. All right, so cells passing through the cranial portion of the streak will form specifically how is this cardiogenic area made up? The cells that pass through the cranial part of the streak form the great vessels. So who are the great vessels? Superior vena cava, inferior vena cava. Some authors leave this one out and don't include that as great vessel. Some do, some don't. We have a pulmonary trunk. We have the very important, behind the pulmonary trunk, we have the ascending aorta, great vessel. And then we have these pulmonary veins, 4 of them coming in the left atria. And then we have pulmonary arteries going away from the right ventricle there. So those are the great vessels. All right, then we have cells that pass through the middle portion of the streak. They form the heart's ventricles. And then cells that pass through the caudal portion of the streak and go forward, they form the heart's atria. All right, and there's a picture of all that. So that's going to form the great vessels. Sometimes that's called the outflow tract. If you hear outflow tract, that means great vessels. And the ventricles are this next wave and this last wave. And remember, we've already talked, right, about cells, how they, we've talked in depth about how these epiblast cells move in and then they dive down, right? We've talked about that. That same principle is applying here. So that's the third wave, forms the cardiogenic area, which is going to become the heart. That was the third wave. Now there's a fourth wave that's going to become the diaphragm. So a lot of authors fail to talk about this, but it's really important. This starts at the 4th week, the beginning of the 4th week, and we get a fourth wave of these cells that migrate through the primitive streak. And they stay even a little bit further lateral, but they go way cranial, past everything. These go the furthest cranial. Plus the trilaminar disc is growing in a cranial to caudal direction. So, but they go even further than the cardiogenic area, and they pile up way toward where the head's going to be, and they become something called the septum transversum. Septum transversum. So here they are. The primitive streak is almost gone. So this is probably the last major thing, bunch of cells that is induced by the primitive streak. But here it is. That's the septum transversum. Got it? And that's going to become the diaphragm. Got it? It's going to become the diaphragm. And that will form a separation between the thoracic and abdominal cavity, and that is the diaphragm. The other thing about that is it gives rise to the liver. So the whole liver morphs by certain genes getting turned on in a certain region of septum transversus, and that gives rise to the liver. Specifically, the cells that become the liver start expressing BMP4 and Wnt are the genes that are involved with that. And there's a diaphragm if you don't know what that is. That's again gross 2. This would be helpful if this was probably after gross 2. I've said that like a million times, but it's a dome shape there. We have a tendinous part and a muscular part on the outside. Talk about that more when we talk about GERD in 5th quarter, but yeah, there it is. All right, so now let's talk about something really important. I've talked about these somites, and I keep saying we're going to learn about somites in the future. Well, let's learn about these somites. So the formation of somites is called somitogenesis. Somitogenesis. Some say somerogenesis. I say somitogenesis. All right, and that is the somites come from the paraxial mesoderm. Now we've already talked about that layer that's closest to the midline, the paraxial mesoderm, because it underwent segmentation and clumping. And that's basically what this is, but we're going to get into it a little bit more. And then there's somites as well, but they're not quite as clumped and they're not quite as visible, and they're up in the brain region. Those are called somitomeres. Somitomeres. So let's talk about them. So this process of forming somites is called segmentation. Some authors call it somitogenesis, but segmentation is an a.k.a. Somitogenesis is way more descriptive though. And it's just when the paraxial mesoderm clumps into blocks of epithelial structures called somitomeres or somites. So let's go through it. So about day 19, it starts up in the cervical region. So small region in the cervical most part of the paraxial mesoderm. Remember these cells are epidermal, right? These are still epidermis cells, or epithelial cells. So the first thing that happens is we have to, they're still mesenchymal, right? Because all mesoderm is mesenchyme if you want to clump it together, you have to turn it into epithelium. So the first thing that happens is this cranial part, paraxial mesoderm, undergoes mesenchymal to epithelial conversion. So we won't get into that. But these maybe we will a little bit, but not right now. The new epithelial cells are now bound together by CAM cell adhesion molecules, and we won't get into that. But because they have CAMs pulling them tight, they clump. And you can see these clumps. And they're not as strongly clumped as we're going to see below the level of C7. But they're still clumped. So these are called somitomeres that are up in the cervical and occipital region of the embryonic disc. That clumping process can also, here's Langman again, that darn Langman. So he calls it epithelialization, the only one to call it that. Epithelialization. So there's yet another a.k.a. So segmentation, somitogenesis, epithelialization is all kind of the same thing. Almost immediately after the first pair of these somitomeres clump, another pair form, and we'll see how that works in a minute. And then after they form, another pair form, and we're moving caudally. This formation is from cranial to caudal. That's an important concept. Cranial to caudal. All right, and this happens over and over until we get to 7 pairs have been created. And so you shouldn't call these somites up in the cranial and occipital region. These are somitomeres because they're not segmented as strongly. All right, so now we can see what they kind of look like. So there's our paraxial mesoderm, and it's clumped into these 7 sections. Those are the somitomeres. And the first pair happened up in the occipital region. Did I say the cranial region? It's in the occipital region that this happens. Yeah, and that's what happens. And, oh, no, I was right. They do happen. Now we're going to somites. I see we switch gears here. So after the 7th one has formed, then the first pair of somites develop, and now we're in the occipital region. So we're out of the cranium. This is all considered cranial region. So I know it seems weird that the occiput is way down here, but this is going to fold like crazy. This is going to get pulled way up in here to the head here. But about day 20, our first pair of somites, and they're really just somitomeres. It's the same story, but these clump so strongly that they stick out like a sore thumb. And again, they're going converting from mesenchymal tissue into epithelial tissue. We'll look a little bit about how that happens. But once pair number 1 here of somite forms, immediately another pair forms, and then another pair forms, and this keeps going and going. The somites are much more strongly segmented too compared to somitomeres. Let me get just a little bit of water here. And you can literally, with a good powered microscope, you can see these things. So here is an electron microscope, and you can clearly see that, like, this is probably the start of them right here. Or maybe, yeah, this one right here, because these are kind of, you can see it a little bit, but their somitomeres are not as segmented, but the somites are incredibly. And this is Paraxial Mesoderm we're seeing. And we're kind of seeing it. Remember, there's a layer there's ectoderm over the top of this. So we're seeing them right through the ectoderm and the right below the ectoderm, because this is mesoderm, right? Intermediate mesoderm would be here. Lateral plate mesoderm would still be out here, right? Neuropores, we talked about the neuropores, and neurulation is still going on. The zipping up of this tube and neurulation pretty much follows the somites growth this direction. All right, so new pairs develop sequentially. It's about, you should know this number, about 3 new pairs of somites form per day, to give you an idea of the speed going here. So here's the cranial region. This is the caudal region. This region of where a new somite is going to form is called the presomitic zone or presomitic area. So that's important. So somite number 6 is going to be forming right here in the presomitic area. All right, here's a better picture of it. There's the presomitic area. Right, and again you can see presomitic area would be right here. So what goes on a little bit deeper on these things? We've got to know a little bit of, actually put these slides in just for this quarter. Aren't you guys happy? So retinoic acid, we need to know a little bit about retinoic acid, FGF8 and Wnt, because those are the two, and it goes really into the weeds. On my YouTube lecture I went way into the weeds on this. If you go look at probably a year or 2 ago, I went deep into the weeds in this. But I'm just going to go a little bit into the weeds. I think that this will be plenty for at least chiropractors. So the last pair of newly formed somites, they get some genes turned on. Specifically, the retinoic acid gene gets turned on. And it does what a gene does. It produces retinoic acid via the central dogma. And the retinoic acid then soaks into the presomitic zone below. So these new genes right here, retinoic acid is soaking into this region right here. And it's getting into the paraxial mesoderm cells right here. So what does it do? Well, it turns off some genes that are naturally turned on. These are the growth genes. FGF8 and Wnt, they cause the disc to grow caudally. And so we need to turn those off. So with Wnt and FGF8 off, then a new somite will form when it gets exposed to retinoic acid. Right? That model has a name. It's called the wavefront model. You should know that word, the wavefront model. Okay, according to the wavefront model, the paraxial mesoderm of the embryonic disc has a gradient going on of FGF in one region predominant and Wnt and retinoic acid in another. Or really, FGF and Wnt go together and retinoic acid, I'll show you a picture here in a second, going on. So only when paraxial mesoderm cells are exposed to high levels of retinoic acid can somitogenesis occur. And again, the retinoic acid turns off the FGF8. So here's the classic story of this. So here's the gradient. So retinoic acid is ruling here in the paraxial mesoderm. And nothing's happening out here. This is just saying, well, these are somite cells. They've already went through somitogenesis, and they're cranking out retinoic acid. These last ones crank out retinoic acid, and it spits right into this region right here, and that turns off FGF4 and Wnt. And once that is turned off, they morph into new somites. And the same thing happens. The retinoic acid goes. So bottom line, somitogenesis occurs from cranial to caudal. And you have to turn off FGF and Wnt in order to do that. Got it? Results of segmentation. So we like results. So by week 5, there's approximately 43 somites that have been formed. And then some authors say the first pairs of these, of occipital somites, and the final few pairs, about the 4 pairs of coccygeal somites, disappear. But whatever happens, it gives us a final count, usually of 38 pairs of somites. Got it? We're not counting somitomeres, just somites. Right? So here's just a folding little creature. Nice arm buds there. Let's count 1, 2, 3, 4, 5, 6, 7. So that's the last somitomere. And now we're starting the somite count right there. And they go all the way down into the coccygeal region. So basically here's where the 38 are after all the dust settles. And it could, some say the end of the 5th week, some say the 6th week. Bottom line is we have 4 pairs of occipital somites. We have 8 pairs of cervical somites, which match the 8 spinal nerves. We have 12 pairs of thoracic, which match the 12 pairs spinal nerves. 5 lumbar, 5 sacral. 4 coccygeal. Hey, that matches the spinal nerves, doesn't it? Except coccygeal, there's only 1 coccygeal nerve. Remember, the remnants of that is in the phylum terminale. The remnants of those other 3 make up the phylum terminale. What do somite? Oh, I like this slide. I like forming stuff. You got to know what stuff forms. You make note cards of this stuff. This is really, pull them out when boards come. This is really high yield stuff. So in general, what do somites form? And we're going to get into the weeds on these things. But in general, you could say they give rise to most of the axial skeleton. So that's the spine, the ribs, and the skull are made from somites. What are somites made from? If you want to even go more general. Paraxial mesoderm. What's paraxial mesoderm made from? Just mesoderm. What was the mesoderm made from? Ooh, really from epiblast cells. Remember I said way back in week 3 or 4, those epiblast cells are magical. They make everything. Anyway, I digress. So axial skeleton. The muscles of the axial skeleton are made. These somites are important, aren't they? Most of the dermis is a little tricky. We'll talk about that, but most of the dermis is made by paraxial mesoderm, or go deeper, made by somites, and we'll show you exactly what it's made by in a bit. The dura mater, blood vessels. The axial skeleton there is in blue. That includes the auditory ossicles. Right? So let's get into this a little bit more, because the somite that we just made is now going to differentiate into 3 layers. So let's keep talking about the somites. The first thing that happens to the somite, we just went through this big story with retinoic acid and FGF8, and now let's even our somite, we can differentiate that into 3 layers. So that's what we're doing right now. The very first thing that happens is the Wagon's circle. In the somite, right in the center of the somite, some of the epithelial cells, they are going to become mesenchymal, because in order to move cells, you can't move epithelial cells, right? You can only move mesenchymal cells because they have little arms. And so they become mesenchymal and they circle. They don't leave the somite. They're going to leave the whole somite in a little while, but for right now they just circle. And these are called somitocele cells. That's what our chiropractic board books call them too. Somitocoel cells. All right. Why do we care about these somitocoel cells? Well, because they, especially with chiropractic, they're some very important structures for us. They will ultimately morph into the annulus fibrosus of the disc. So that's really important, right? How about this? The Z joint. I mean, those are the two main causes of chronic back pain. Tears within the annulus fibrosus and damage to the Z joints, the articular surfaces of the Z joints. They're both filled with pain fiber. And these little covered wagon cells, the somitocoel cells, they form these important structures. Nucleus pulposus, remember, if you're remembering, well, who forms the nucleus pulposus? Most notochord contributes to the nucleus pulposus, as we said, but there's some other things that contribute to that as well. We'll look at. So here's the somitocoel cells, and the hole that they form is called the somitocoel. That's simple, right? So now we got a darn donut right in the middle of our beautifully created somite. So let's further subdivision. So now that the donut is formed, more stuff is going to happen. So the first thing that forms is a very, very important structure called the sclerotome. Sclerotome, you got to know that word sclerotome. You know dermatome, you know myotome from, or you will know those from your diagnostic testing, neurological testing. Sclerotome gets overlooked, and it's really, really important. And it's the first thing that forms. So there's two signaling molecules responsible for this. Sonic hedgehog and noggin are turned on in these cells that are going to become what cells? Specifically, if you're looking at a cross-sectional view or kind of a head-on view or an axial view, the ventral medial portion of our somite, the sonic hedgehog and noggin genes get turned on. Those genes activate PAX1 and PAX9. So this sequence is what makes sclerotome cells, and they're going to become mesenchymal. Parax1 or PAX1, did I say Parax? It's PAX1 and PAX9. So these gene products create another transformation. Well, what the heck? We just turned mesoderm into ectoderm to clump to create the somites, and now we're converting it back into mesenchymal tissue, and that's, yeah, it's crazy. It's crazy. I mean, if you look at the whole picture, remember epithelial cells or epiblast cells were converted into mesenchymal cells that formed the mesoderm, and then we had to clump the mesoderm to form somites. So they went back into epithelial cells, and now we're going back into mesenchymal cells. It's crazy, isn't it? And that's because of PAX1 and PAX9. And you can call this now a secondary mesenchymal tissue because it's already been mesenchyme. It clumped into epithelial cells to form a somite, and now it's back into mesenchyme. So that's called a secondary mesenchymal tissue. So these new secondary mesenchymal cells, they create a new region in the ventral medial portion of the somite, and that's called the sclerotome. And the cells that make up the sclerotome are sclerotome cells. So let's morph our little picture. Here's our head-on picture, cross-sectional, axial, whichever a.k.a. you want to use. This is dorsal. This is ventral down here. So ventral medial, right? This is the midline of the body right here. There's the notochord. And by this time it looks like the neural tube is formed. But this is definitely medial. So dorsal medial, we have these little, you can see the little arms in them. So these have the ability to move, and they're going to move like crazy here in a minute. So these are the new sclerotome cells, which make up the sclerotome. Got it? What do they become? What do they do? This is always a super important question. So they began producing two new substances. Well, not so new. You actually know, but new for the body. The body hasn't seen these before. Called, they're basically making proteoglycans. So these cells turn on genes which make chondroitin sulfate, which is a glycosaminoglycan we talked about in spinal anatomy. and core proteins. So if you have a core protein and you have a gag, they hook up together, and now we have a weird little proteoglycan type molecule that is formed. Well, why would we want a proteoglycan in the middle of these mesenchymal cells? Because proteoglycans will house the next type of cell that is formed from this mesenchymal tissue. So some of the mesenchymal tissue morphs into osteoblasts. Oh, that gives rise to bone, doesn't it? Yeah, and these osteoblasts are actually going to form vertebrae and ribs. We have a little more to the story to talk about first, but that's ultimately where we're going. Fibroblasts and chondrocytes are also created from our mesenchymal tissue, and that's going to give rise to the meninges, right? The dura, arachna, and pia mater. So what's going to happen is our cells are going to take off and divide through mitosis, and they're going to start to, this half of the sclerotome is going to migrate, and it migrates up here to the neurotube, which just forms, and it forms a coating around the neurotube, right? And it forms a coating around the notochord here. That's the neurotube. Notochord. Just put an N for notochord. Right? And this is going to ossify. Then chondroitin sulfates are formed in here, and proteoglycans, and then osteoblasts will get in here. Let's make them red. Osteoblasts will start, and this is, super small, right? I'm making this way bigger than it is. Osteoblasts will start forming here, and we're going to start laying down bone here to create a vertebrae. Isn't that cool? Okay, anything else I need to say? I don't think I need to say anything more there. Here's another classic model. It's important to understand that two adjacent somites, this is a high yield test board question right here. So here's a somite right here. We could pick it, let's say, I don't know what number, how about T5. Okay, that's T5. Here's a T6 somite. And notice that the bottom half of the somite, specifically the sclerotome cells in this region, will migrate and make up this part of the body. And the upper half of T6 will migrate, just like I showed you, around the neurotube, and they'll make up the lower part of the body. And then the sclerotomes also form the disc along with the notochord, remnants of the notochord. So kind of the middle region forms the disc. So that's super important to understand that. So a vertebral body was actually made by two sclerotome units. Okay, or you could say two somite units if you want, because this is just a part of the somite, right? We're splitting up the somite. Does that make sense? And I know it's confusing, but just play it back, listen to it again. So that is the story with the sclerotomes. Sclerotomes are going to make the vertebrae. They're also going to make ribs. That's the star of the show. That's the main function of them. But we still have our somite. We only morphed maybe a third of the somite into a mesenchymal tissue. We still have 2/3 of the somite is still epithelial tissue. So we need to fix that problem. Or we don't need to fix that, but what happens next is we're going to form what's called the dermatomyotome, or the dermomyotome is how they say it, dermomyotome. And so now the dorsolateral half of the somite is going to get stimulated by Wnt gene products. Let's just leave it at that. The Wnt gene stimulates PAX3 and 7 and Paraxis gene to turn on. So with these genes turned on, you're going to make the dermatomyotome. But here's the key. It only happens at the extreme ends of the dorsolateral half. So the middle portion of the dorsolateral half is not going to be affected by this. Right? So let's see a picture of this. So this is what I mean. So PAX3 and 7 and Paraxis only get turned on here. Because now we've already talked about the medial dorsal portion. Now this is the lateral the medial ventral portion. The lateral dorsal portion clumps into mesenchymal tissue, but the middle of it never clumps. It never turns into mesenchymal tissue. We're not exactly sure, don't quite understand how that happens, but we know that PAX3 and 7 and Paraxis genes are turned on only at the ends. So because this tissue can migrate, take a guess what it's going to do. It's going to migrate underneath this epithelial tissue of just the original somite cells. And we're going to form a 3-layer structure is what we're going to do. Okay, and that's everything I said. And it's going to form 3 layers. It's basically these cells, this dermatomyotome is short-lived. It will soon become a dermatome layer, and these epithelial cells will become, these will become the dermatome cells, these epithelial cells, which nothing really happened to them. These cells that underwent epithelial to mesenchymal transformation, they're going to migrate under and become the myotome cells. Okay, so that's just what I said. So all the mesenchymal cells on the end of the dorsal lateral part of the somite, they migrated beneath to become the myotome. The dermatome stayed on the middle. So let's see a picture. See how that's happening? Those cells are moving right underneath. Okay, so cool. So now we have this. And while this was happening, the donut cells in the middle became fully mesenchymal, and they got the heck out of there. They migrated just and kind of mixed in with these sclerotome cells. So they got out of Dodge, so to speak. So the dermatome is still epithelial cells, but myotome and sclero, those are mesenchymal cells. That could be a nice question, couldn't it? Dermatome, what does it give rise to? Oh, these are important, aren't they? The dermatome gives rise to the dorsal dermis of the trunk. So the low back and thoracic region that you're going to be working on as chiropractors, that actually was derived from the dermatome. I mean, we could go shallower though. I mean, we're getting into the weeds when we see dermatome. We could say it was derived from a somite. That's true. We could say it was derived from Paraxial Mesoderm. That's true. We could say it was derived from Mesoderm. That's really too general, but that's all true. See how that works? So now, if the dermatome cells give rise to the dorsal dermis of the trunk, what gives rise to the anterior and lateral dermis of the trunk, and dermis of the limbs for that matter? It's lateral plate mesoderm in general. And remember, we said that we could get even deeper than that if we wanted. But for now, we'll just leave it lateral plate mesoderm. All right, central region of the dermatome also gives rise to, so that dermatome region, cells in the very center. This is new. I don't think they're going to ask this on boards, but it does give rise to some skeletal muscle and some brown fat as well. What about the somitocoel cells? I told you they got out of Dodge. They became fully mesenchymal and crawled out of this evolving story here, and they're hanging out over here. Some authors call these, they don't call them somitocoel cells anymore. Some authors call them arthroptome cells at this point. Arthroptome cells. Chiropractic board books, though, they just continue to call them somitocoel cells. Right? What do they become again? Well, we said they give rise to the annulus fibrosus. Some important stuff, right? They give rise to the annulus fibrosus of the disc. And remember, nodal cord cells, they also make up the nucleus pulposus. And we looked at some other cells that contributed to that as well. Basically, the sclerotome also makes up the nucleus pulposus. But somitocoel cells make up the annulus fibrosus, super important. They make up joint surfaces, specifically the Z-joints, articular pads, or the facets of the superior and inferior articular processes, made by these important covered wagon cells, somitocoel cells. They also make up the proximal part of the ribs. What does the sclerotome give rise to then? Well, we already talked about that, but I want to go into the weeds on this. Oh yeah, let's get into the weeds. So we can actually, the research is so good in Carlson, we know well, we already know what the sclerotome becomes, but we can break it down even further. So the ventral, and remember the sclerotome, don't get that confused with the somite. Because remember the sclerotome, this is the somite, right? Here's the neurotube, there's the nodal cord. There's another one over here on this side. So remember we said this region right here becomes sclerotome, right? So the ventral region, that would be this region down here. The ventral region of the sclerotome actually is what really gives rise to the vertebral bodies and the intervertebral discs. That's where those cells come from. Remember, it takes 2 sclerotomes to make a vertebral body. The dorsal region, well, this would be more dorsal right up here. The dorsal region gives rise to the spinous processes in the lamina. The lateral region, which would be over here, cells, mesenchymal cells, morph into the distal ribs and some of the tendons and some of the intrinsic back muscles, like the multifidi muscles. The medial region, which is all what we have left, or now we have the central region left, medial region will be right here. They form the superior and inferior articular pillars, or articular processes. They don't form the facets though, or the facets on the top of those. They form the bone. They form pedicles, transverse processes, and the proximal ribs as well. Or no, not yet. The center process would be right here. They form the superior and inferior articular processes, the pedicles, transverse processes, and ribs. That medial portion only forms the meninges, right, and some of the blood vessels of the meninges as well. That medial region, this region right there, is sometimes called the meningiotome. Meningiotome. This lateral region out here is sometimes called the syndiotome. Syndiotome. Got it? Another really important slide. All right. Myotome. What does that layer give rise to? Well, it must be muscle. Myo means muscle. Well, we can split that into 2 parts as well. So the medial or the dorsal medial part of that gives rise to the rest of the intrinsic back muscles and the extensor muscles of the limbs. Who are these intrinsic muscles of the back? It's every back muscle that you're going to learn next quarter, except for the big muscles, the trapezius and the latissimus dorsi. Those are extrinsic muscles. Everything else is intrinsic muscles. The ventrolateral portion of this tissue, it's probably better to just say lateral, because we've got folding going on now. So the lateral portion gives rise to the muscles of the ventrolateral body wall. So those are the flat muscles, which you're going to learn next quarter. So who are the flat muscles? Those are the side muscles. Those are the external oblique, internal oblique, transversus abdominis muscle. It also gives rise to the erectus abdominis, and there's a little baby muscle called pyramidalis. It's usually dissected away. Mama Z always gets rid of that one. But there's some fiber there. It's like a mini erectus abdominis muscle. And then the flexor muscles of the limb are also created by myotome. Here's the flat muscles. We'll look at this when we talk about hernias. It's the same picture in 5th quarter. But there's the external oblique muscle, internal oblique, and then the inner one there is transversus abdominis muscle. Then we got this important fascia. I'll try not to digress. The rectus abdominis muscles are here, right? Tendinous intersection is here. What else? Linea alba is here. We can have hernias around here. Linea semilunaris, you should add that to your gross anatomy studies. Has she added that yet? I doubt if she's added that, but that's really important because that's where a Spigelian hernia occurs. Spigelian hernia occurs in the linea semilunaris. Anyway, I'm digressing. All right, so there's our mature somite. Are we done? I think we're done. No. What else can somites make? Just a little cleanup stuff. So somites, I mean, they're stem cells. They can morph into many different types of tissue. All of the somite tissue and cells, all that mesenchymal cells, they can morph into blood vessels in a process called angiogenesis. So specifically, here's the list that somite cells can morph into. Fat cells, adipocytes, chondrocytes, osteoclasts, endothelial cells, which is the inner lining of the blood vessels, pericytes, which are down in the venules, which you probably learned in histology, fibroblasts, myocytes, so smooth muscle cells, and skeletal muscle cells, and nervous system cells. So I'm just scratching the surface. These somite cells do a lot more than make muscle and bone and things like that. All right, so I hope you learned a lot this quarter. This stuff is hit on boards pretty good, so make sure if you know this stuff, you're going to have to do a little extra study because we don't have a part 2 of this class, what we need. But you'll certainly, when you take your board review class, you'll certainly understand it a lot more when you start, because really, like, making the respiratory system is so easy compared to all this stuff. All right, anyway, we'll see you guys in 5th quarter.